Good morning children. Welcome back to Botany class today. Children, in the previous class we all learned about genetics, isn't it? What is genetics? The branch of biology that deals with genes, genetic variation and heredity is known as genetics, isn't it mom? Who is called father of genetics? Greher Johann Mendel. Greher Johann Mendel is called father of genetics. Okay, which plant he selected for this experiments? Garden pea plant. You know the 7 pairs of contrasting characters of pea plant, isn't it? The pea plant possesses 7 pairs of contrasting characters. Okay, what is called monohybrid truss? Crosses involving the inheritance of only one pair of contrasting character is called monohybrid cross. What is the phenotypic ratio of monohybrid cross? 3:1. What is the genotypic ratio of monohybrid cross? 1:2:1. What is called phenotype? The external expression of a particular organism is called phenotype. The genetical expression of a particular organism or genetical expression of an organism is called genotype, isn't it mom? So today we are going to learn about dihybrid truss. Okay, so dihybrid truss. The crosses involving the inheritance of 2 pairs of contrasting characters are called dihybrid cross. Okay, 2 pairs of contrasting characters chosen by Mendel are shape and color of seeds. One is the shape, another is the color of the seeds. These 2 characters he chosen for this dihybrid cross. Okay, so he selected round yellow seeds and wrinkled green seeds. Okay, round yellow seeds and wrinkled green seeds. Okay mom, wrinkled green seeds. This round yellow seeds are represented by capital R, capital Y, capital Y. Green wrinkled seeds are represented by small r, small y. These are the parents, pure breeding parents. Okay, and he crossed pea plants having pure breeding round yellow seeds with pure breeding wrinkled green seeds. In F1, he found only the round yellow seeds. Okay, in the what is F1? First generation. He found only yellow round seeds. Okay, no green wrinkled seeds are appeared in the F1. Okay mom, so from this he concluded that the round yellow seeds are dominant over wrinkled green. Listen, here this round yellow seeds produce the gametes like this, capital R, capital Y, here small r, small y. These are the gametes, isn't it mom? These gametes when fused to form the F1 hybrid, that is capital R, capital Y, small y. This is F1 hybrid or first generation, isn't it? F1 hybrid or first generation. This pea plants having round yellow seeds crossbred by self-pollination. The 4 types of seeds having different combinations of shape and color were obtained. Okay, this is F1 hybrid. These F1 hybrids, the externally or phenotypically it is round yellow, isn't it? Round yellow. These crossbred or by self-pollination. These F1 hybrid crossbred among themselves. They produce 4 types of seeds having different combinations of shape and color. Okay, in the F2 generation or second generation. You have to write the hybrids like this. This gamete, from this hybrid we have to write the gametes. Okay, capital R, capital Y, then small r, capital Y, then capital R, small y, small r, small y. Okay, these are the gametes from F1 hybrid. When these F1 hybrid crossbred among themselves, these produce 4 different types of seeds, isn't it? So we have to write the this gametes like this, capital R, capital Y, then small r, capital Y, capital R, small y, small r, small. The same gametes you have to write here. So for F1, this we have to note down the F1, you have to draw the checker, but it's a graphical representation, isn't it? To note down the hybrids. So this gamete, first this gamete fused to form, these gametes fused to form, these gametes and form this round yellow seeds. Okay, so R, capital R, capital R, capital Y, capital Y. Write down here. Then this gametes fuse with that gamete and form their respective hybrid. Capital R, small r, capital Y, capital Y. Then this gamete fuse with that gamete and form their respective hybrid. That is capital R, capital R, capital Y, small y. Then here this gamete fuse with that gamete and form their respective hybrid. Okay, so here also this gametes fuse all the gametes and write down the hybrids here. Capital R, small r, capital Y, capital Y. Then this gamete, this hybrid, how to write is small r, capital Y, capital Y. Then here you have to write these two, fusion of these two gametes results in the formation of this hybrid. Then here these two you have to write down here. Okay. Then capital R, capital Y, small y. Capital R, small r, capital Y, small y. Write down here. Then here these two hybrids, these two gametes fused to form this hybrid. Capital R, small y, small y. Next, capital R, small y, small y. Write down here. Then this gamete fuse with that gamete and form their respective hybrids here. Capital R, capital Y, small y. Then small r, capital Y, small y. Here, capital R, small y. Write down here. The last hybrid, this one, small r, small y, small y. These, so you have to write down the hybrids here. There are from this the ratio phenotype of seeds in the F2 generation is 9:3:3:1. This is a yellow round. 1, 2, 3, 4, 5, 6, 7, 8, 9. These 9 hybrids externally shows yellow round. Okay, so 9 hybrids are yellow round. Then yellow wrinkled 1, 2, 3. Okay, these 3 hybrids are yellow wrinkled. Then green round 1, 2, 3. These are the green round hybrids. Green wrinkled, this one. Okay, so 4 different types of hybrids are formed in the F2 generation. Okay, ma'am. How many different types of hybrids are formed in the F2? 4 different hybrids are formed in the F2. Okay. The ratio of phenotype in the seeds in F2 generation is 9:3:3:1. 9 with the 2 dominant traits. This dominant character, this also dominant. Dominant. So 9 hybrids with the dominant character. Then 3 with 1 dominant and 1 recessive. Okay. These 3. This is recessive character and 1 dominant character. Like this, 3 hybrids are hybrids having 1 dominant and 1 recessive character. Then 3 with another dominant. Here this is here 1 dominant, yellow dominant. Here this round is dominant. So this another 3 with another dominant and another recessive. Okay, another 1 is the recessive. 1 hybrid shows the recessive character. 2 recessive character, that is green wrinkled. Okay, that is one of the recessive. One with 2 recessive character. So 9 hybrid with 2 dominant traits, 3 with 1 dominant and 1 recessive, another 3 is with another dominant and another recessive gene, another recessive traits, then only one, this one only, where 2 recessive characters. So these are the 4 types of plants are formed in F2 generation. Okay, two new combinations are formed, that is round green and wrinkled yellow. Okay, round green and wrinkled yellow. These two are the new combinations of traits formed in F2. Okay, so from this experiment, based on monohybrid and dihybrid crosses, Mendel stated 3 laws. Okay, first law is law of dominance, second one is law of segregation, and third one is law of independent assortment. From monohybrid and dihybrid crosses, Mendel stated 3 laws. What are these laws? Are called Mendel laws of heredity. First law, law of dominance. When two homozygous individuals. What is homozygous? When two factors are same. Okay, this is homozygous. Isn't it? Two factors are same. When two homozygous individuals with one or more sets of contrasting characters are crossed, the characters that appeared in F1 generation. The F1 generation is dominant. The character appeared in F1 generation is dominant, and those do not appear in F1 are recessive. You already know that what is dominant character. The character which is expressed out as dominant, a character which is masked or hidden, that is recessive, isn't it? So law of dominance, this law states that when two homozygous individuals, these two are the homozygous individuals, isn't it? The factors are same. Homozygous individuals are crossed with one or more pairs of contrasting characters, then the characters that appear in the F1 are dominant, and the character which is masked or which is hidden, that is recessive. Okay, so that law is called law of dominance. What is the first law? Law of dominance. Second law, law of segregation or law of purity of gametes. Okay, when a pair of contrasting characters or factors or alleles are otherwise said to be allelomorphs, these are brought together in a heterozygote or hybrid. This is heterozygote or hybrid. Why it is impure, isn't it? When a pair of contrasting characters or genes or allelomorphs are brought together in a heterozygote or a hybrid, the two members of the allelic pair remain same. When gametes formation, these will separate. Okay, these are the gametes, isn't it now? When gametes formation, these factors will separate. Okay, that is called law of segregation or law of purity of gametes. Okay, till that they remain together without mixing. When gametes formation, these factors are separate. So in the before that, the heterozygote, the factors will remain together, or allelic pair remain in a heterozygote or in a hybrid. Then when it is separated at the time of gamete formation, that is law of segregation, or it is termed as law of purity of gametes. Third law, law of independent assortment. Okay, in case of inheritance of two or more pairs of contrasting characters, two pairs of contrasting characters or simultaneously, this the factors of one pair assort out independently. Okay, that is called independent assortment. Inheritance of two or more pairs of contrasting characters or contrasting characters simultaneously, they are in a zygote, the factors of one pair are sort out independently of the other pair. That is independent assortment. Now we are going to learn about chromosome. Okay, you all know that human body is made up of millions of cells. The nucleus of each cell contain thin thread-like structures called chromosome. So in the cell, chromosome. What are chromosomes? What are chromosomes? The nucleus of each cell contain thin thread-like structures called chromosomes. Okay. Chromosomes, the term chromosome was first introduced by Valdeir. Okay, mom? So chromosomes in 1888. It was first coined by Valdeir. Who coined the term chromosome? Valdeir. In 1888. The chromosomes are highly condensed structure which is packed with the chromatin fibers or with the DNA. What is DNA? Deoxyribonucleic acid. That is DNA. Now this chromosomes are highly coiled chromatin fibers packed with the DNA. These chromosomes are the physical carriers of genes. These genes are the segments of DNA responsible for a particular phenotypic character. The gene is present in the chromosome. For example, in the chromosome is having the arms called chromatids. In the chromosome, genes are located. Okay. So this the specific the genes are segments of DNA responsible for a particular phenotypic character. Each gene is present in the chromosome in a specific portion that is called locus. Okay. So each gene is present in the specific position in the chromosome that is called locus. Now the structure of chromosome. The chromosomes are thin, long and thread-like structures containing two identical structures called chromatids. Okay. So chromosomes are thin, long, thread-like structures consisting of two identical structures called chromatids. These are the chromatids. These two chromatids are joined by a central body called centromere. What is called centromere? The chromatids are joined or two identical strands of or sister chromatids are joined in a point called centromere or the central body. Okay. So each chromatid is made up of spirally coiled. This is the structure of chromosome. Each chromatid is made up of spirally coiled structure, thin structure that is called chromonema. What is it, ma'am? Chromonema. Chromosome has number of bead-like structures. Okay, number of bead-like structure that is called chromonemata. The bead-like structures are called chromonemata. Chromosomes are made up of DNA, RNA, proteins and metallic ions. Okay. These proteins provide the structural support to the chromosome. The chromosome consists of four important parts. The outer covering of chromosome is called pellicle. What is pellicle? The outer covering of the chromosome is called pellicle. A chromosome consists of four important parts. One is primary constriction. The two arms of chromosomes are joined by a central body. It is called the primary constriction or it is centromere. Okay. The two arms of chromosomes meet at a point called primary constriction or centromere. The centromere is a region. This is the region where spindle fibers attach during cell division. Okay, so primary construction is also called centromere. This is the region where spindle fibers attach during cell division. Next, secondary construction. Some chromosomes possess secondary construction at any point of its body. Then there are these regions are called nucleolar organizer. Okay, the secondary construction. These are also called nucleolar organizer. This nucleolar organizer helps in the formation of nucleolus in the nucleus. Next, telomere. The end of the chromosome is called telomere. The telomere helps it helps to prevent it from joining the adjacent chromosome, that is telomere. It maintains and it provides the stability to the chromosome. Next one is satellite, chromosomal satellite. These some of the chromosomes have elongated knob-like appendages at one end. This is called chromosome. This is called chromosomal satellite. The chromosomes having satellites are called chromosome. Okay, these chromosomes having satellites are called chromosomes. This is the structure of chromosomes. So chromosomes are thin, elongated, rod-like structure present inside the nucleus of the cell. Now, you all know that human body is made up of millions of cells. The nucleus of each cell contains the thread-like structures called what? Chromosomes, isn't it? Who introduced the term chromosome? Valdeir in the year 1888. The chromosomes are the carriers of genetic material which are transmitted from one generation to the next generation. The chromosomes are thin, long, elongated structure. Thin, long, elongated structure consisting of two identical structures called what? Sister chromatids. These sister chromatids are connected by a central body called what? Centromere. Okay. Then each chromatid is made up of spirally coiled thin structures called chromonema. Spirally coiled thin structures called what? Chromonema. These chromonema contain bead-like structures called chromonemata. Okay, the chromonema has the number of bead-like structures called what? Chromonemata. The outer covering of the chromosome is called what? Pellicle. Okay, pellicle. Then this chromosome has number of bead. The chromosomes are made up of DNA, RNA, proteins. The proteins are histones and non-histone proteins. Which one provide the structural support to the chromosome? Proteins. Proteins provide the structural support to the chromosome. What are the parts are there in chromosome? What are the four regions? Primary construction, secondary construction, telomere and satellite. Okay. The primary construction, that's also called centromere. Some of the chromosomes have the secondary construction. Okay, what is the function of primary construction? This is the region where spindle fibers are attached during cell division. Okay, it connects the two arms of the chromosome. Next, secondary construction. It is also called nucleolar organizer. Helps in the formation of nucleolus in the nucleus. Yes, then telomere. It prevents the chromosome from adjoining the adjacent chromosome. Okay, it maintains, the telomere maintains and provides stability to the chromosome. Satellite, what about satellite? Some of the chromosomes have knob-like appendages, that is called satellite. This chromosome having satellite are called chromosomes. Okay, the chromosomes having satellite are called chromosomes. These are elongated knob-like appendages found at the end of the chromosome. Telomere is the terminal part of the chromosome. Okay, children, you have to learn these topics. Now we all turn page 271. Turn page 271. Second, choose the correct answer. Second one, mark it. 9 is to 3 is to 1. 9 is to 3 is to 3 is to 1. Ratio is due to independent assortment. Number 3, the region of the chromosome where spindle fibers get attached during cell division, centromere. Isn't it, ma'am? Fill in the blanks. Third one, the thin thread-like structures found in the nucleus of each cell are called chromosome. Roman number 3, identify whether the statement are true or false. A typical Mendelian dihybrid ratio of F2 generation is 3 is to 1. What is the ratio of dihybrid cross? 9 is to 3 is to 3 is to 1, isn't it? So, the given statement is false. You have to write false and underline 3 is to 1, and instead of 3 is to 1, what you have to write is 9 is to 3 is to 3 is to 1, okay? 9 is to 3 is to 3 is to 1. Then second one, a recessive factor is altered by the presence of a dominant factor. True. Number 3, each gamete has only one allele of a gene. Answer is true. Number 4, hybrid is an offspring from a cross between genetically different parent. True. Number 5, some of the chromosomes have an elongated knob-like appendage known as telomere. That is false, isn't it? Instead of telomere, what you have to write is satellite. Then turn next page. Answer in a sentence. First question, what is a cross in which inheritance of two pairs of contrasting characters are studied? Dihybrid truss, right? Dihybrid cross. Dihybrid cross. Then number 2, name the conditions when both alleles are identical. Homozygous. Homozygous. Number 4, 3 are written already. Number 4, what is the name given to the segments of DNA which are responsible for the inheritance of a particular character? Genes. Next question, Roman number 6, short answer, 6th one. A pure tall is crossed with a pure dwarf plant. What will be the F1 and F2 generation? Explain. Page 262. Answer is there in page 262. Under the heading, monohybrid, Mendel's explanation of monohybrid truss. From parental generation, you have to mark up to 3 is to 1. Up to the heading, F2 generation, first 3 lines. To the ratio of 3 is to 1, which write down a sentence there, which is the phenotypic ratio. Selfing of F1 monohybrids resulted in tall and dwarf plants respectively in the ratio of 3 is to 1, which is the phenotypic ratio. Okay, the next paragraph, in the up to that you mark it, then come to the next paragraph. In the F2 generation, 3 different types were obtained. Tall homozygous, capital T, tall heterozygous, capital T, small t, 2, dwarf homozygous, small t, 1. Up to that you mark it. Okay. Then question number 7. Explain the structure of chromosome. It is there in page 265. Explain the structure of chromosome. Page 265, the last paragraph. The chromosomes are thin, long, from there you mark it, up to SAT chromosomes. Up to SAT chromosomes. Next question. Long answer question. Explain with an example the inheritance of dihybrid truss. How is it different from monohybrid truss? Answer is there in page 263 and 264. 263. Below the heading Dihybrid Truss, you have to mark it. Dihybrid Truss involves from there up to the next page, first two paragraphs up to dihybrid ratio. Roman number 7.1. One more question is asked. How is it different from monohybrid truss? You have to write down the answer for this question. Draw two columns. Monohybrid truss. Monohybrid truss. Draw two columns and write. Okay. Under the heading monohybrid truss, first point, monohybrid truss is a genetic cross. Genetic cross that involves a single pair of genes which is responsible for one trait. Which is responsible for one trait. Second point, parents differ by a single trait. Parents differ by a single trait. Third point, monohybrid ratio in F2 generation is 3:1. Next heading, dihybrid truss. First point, dihybrid cross is a genetic cross that involves two pairs of genes which are responsible for two traits. Second point, the parents have two different independent traits. The parents have two different independent traits. Third point, the dihybrid ratio in F2 generation is 9:3:3:1. Okay children, you have to learn and write the long answer. Roman number 7.1. Roman number 7.1. Short answers 6 and 7. Learn and write in your notebook. Thank you children. We shall